1918 witnessed Ronald Aylmer Fisher's seminal work, The Correlation Between Relatives on the Supposition of Mendelian Inheritance, a publication that effectively ended a significant scientific conflict. Early 20th century biology grappled with a profound division. The Biometricians, led by Carl Pearson, meticulously quantified continuous variation in traits using correlation and regression. The Mendelians, staunch advocates of Mendel's discrete, particulate inheritance, struggled to reconcile their findings with the Biometricians' data. Fisher's paper provided a masterful mathematical bridge, resolving this seemingly intractable conflict. The central challenge involved reconciling continuous traits, such as height, with Mendel's discrete gene inheritance. Fisher ingeniously proposed the multiple-factor or polygenic hypothesis. Numerous Mendelian factors, each contributing a small effect, could collectively generate the continuous variation observed. This elegant solution rested on a novel mathematical framework, the Analysis of Variance ANOVA. Fisher partitioned total phenotypic variance VP into its constituent parts. He identified additive genetic variance VA, representing the sum of individual gene effects, dominance variance VD, reflecting deviations from simple additivity due to dominance interactions, and environmental variance VE, accounting for non-genetic influences. He also considered the impacts of assortative mating on variance components. Fisher's mathematical brilliance is showcased in his derivation of theoretical correlation coefficients for relatives under this polygenic model. He predicted a correlation of 1/2 for parent offspring and full siblings, and 1/4 for half-siblings. These theoretical predictions closely matched empirical data collected by the Biometricians, providing compelling evidence for his model. Critically, Fisher treated dominance not as a separate phenomenon, but as a deviation from the additive model, a crucial insight for his variance partitioning. This approach allowed him to incorporate dominance effects within the ANOVA framework, further strengthening his model's explanatory power. The paper explicitly depended on Pearson's statistical methods and data, providing the empirical framework for Fisher's analysis. Furthermore, the Mendelian principles of segregation and independent assortment constituted the fundamental suppositions underlying Fisher's model. The paper's success stemmed from its precise mathematical framing of a biological problem. It meticulously defined variance components, carefully derived correlation coefficients, and directly compared theoretical predictions to observed data. The resulting near-perfect correspondence offered strong support for the multiple-factor hypothesis. Fisher's ANOVA method, while initially developed for this specific problem, rapidly transcended its origins. Its utility extended beyond the study of relative correlations, becoming a ubiquitous statistical tool across diverse scientific disciplines. His work at Rothamsted Experimental Station immediately demonstrated the practical power of ANOVA in agricultural research, optimizing crop yields. Later, Sewell Wright and GWAS Halding built upon Fisher's framework, extending its applications to broader evolutionary contexts. Wright's path coefficient method, arriving at conclusions similar to Fisher's, sparked a productive, albeit often contentious, intellectual relationship. Haldane, meanwhile, applied Fisher's mathematical insights to model gene frequency changes under selection pressure. The legacy of Fisher's variance partitioning approach extends to the modern concept of heritability, particularly narrow since heritability H2 via VP. This ratio, directly derived from Fisher's 1918 formulation, remains fundamental to animal and plant breeding, and is central to understanding complex traits in human genetics, informing studies such as genome-wide association studies GWAS. A century of genetic research has overwhelmingly corroborated the core claims of Fisher's 1918 paper, solidifying its status as a foundational text in quantitative genetics and a cornerstone of the modern evolutionary synthesis. Fisher's achievement was nothing short of transformative. A relatively unknown schoolteacher's theoretical work provided the mathematical engine for a profoundly new understanding of evolution, solidifying evolutionary biology's transition into a truly quantitative and predictive science. The 1918 paper's true legacy lies in its role as a genesis event. From its mathematical architecture sprung the entire discipline of quantitative genetics, providing the conceptual tools that would underpin the modern evolutionary synthesis. The paper's publication was akin to striking a match in a room filled with intellectual kindling. Almost immediately, its influence permeated the work of other brilliant minds grappling with the same fundamental questions of heredity and evolution. Across the Atlantic, the American geneticist Sewell Wright was developing his own powerful method of path coefficients. This graphical and algebraic technique, while conceived independently, arrived at remarkably similar conclusions regarding the correlations between relatives. The convergence of their results initiated perhaps the most significant and enduring intellectual rivalry in 20th century biology. For decades, the Fisher and Wright debate over the relative importance of genetic drift versus natural selection, fueled by their differing mathematical frameworks, would sharpen the field's thinking and push evolutionary theory to new heights of quantitative rigor. Simultaneously, the polymath GWAS, Haldane seized upon the new mathematical possibilities. He extended Fisher's statistical approach, building a series of seminal papers throughout the 1920s that modelled the rate of change in gene frequencies within a population under the force of natural selection, where Fisher provided the static framework for understanding variance at a single point in time. Haldane set that framework in motion, creating the first robust mathematical treatment of evolution in action. Together, the triumvirate of Fisher, Wright, and Haldane formed the intellectual core of population genetics, each building upon the foundational possibility demonstrated in 1918 that the complexities of evolution were amenable to precise mathematical description. Their collective work established the theoretical bedrock for the synthesis of Darwinian selection with Mendelian genetics, a unification that had appeared impossible a mere decade earlier. Beyond its specific biological conclusions, the 1918 paper contained the seed of a universally applicable statistical tool in partitioning the sources of phenotypic variance into their genetic and environmental components. Fisher had laid the conceptual groundwork for what he would later formalize as the Analysis of Variance, or ANOVA. This ingenious method for comparing the means of multiple groups by analyzing their variance became one of the most powerful and widely used statistical innovations in history. Its immediate impact was felt most profoundly at Rothamsted Experimental Station, the world's oldest agricultural research institution, where Fisher began working in 1919. At Rothamsted, Fisher applied ANOVA with devastating effect to the messy, complex data of agricultural field trials. For generations, agronomists had struggled to determine whether differences in crop yield were due to a new fertilizer, a different planting technique, or simply random variations in soil quality across a field. ANOVA provided the solution, a rigorous method to disentangle the variance attributable to the experimental treatments from the variance attributable to random error. It allowed researchers to state with a calculated degree of certainty whether one fertilizer truly produced a superior yield over another, revolutionizing experimental design and agricultural science. A technique born from abstract questions about human inheritance found its first world-changing application in the cultivation of wheat and potatoes, a testament to the profound utility of Fisher's mathematical mind. The deep logic of variance partitioning from the 1918 paper echoes directly into the central concepts of modern genetics. The very idea of heritability, a measure of how much phenotypic variation in a population is due to genetic variation, is a direct intellectual descendant of Fisher's work. Specifically, the concept of narrow-sense heritability is a precise restatement of his core insight. This value, represented as h², is calculated as the ratio of additive genetic variance, denoted V sub A, to the total phenotypic variance, denoted V sub P. This ratio quantifies the proportion of observable differences among individuals that is transmitted to their offspring. It represents the component of inheritance upon which natural selection, or artificial selection, can effectively act. This single, elegant concept remains the cornerstone of modern animal and plant breeding programs. Livestock breeders use estimates of heritability to predict the response to selection for traits like milk production in cattle or lean muscle mass in swine. Its principles guide the development of higher yielding and more resilient crop varieties. In human genetics, this same foundational logic underpins massive endeavors like genome-wide association studies, or GWAS, which seek to identify the numerous genes of small effect that contribute to the variance of complex traits and diseases. The mathematical engine Fisher, built to understand familial resemblance a century ago, now powers our search for the genetic architecture of human health itself. The 1918 paper represents a singular moment of scientific transfiguration. It was a work of pure theory that resolved a bitter and protracted biological dispute, providing the mathematical conciliation between the Biometricians and the Mendelians. Yet its achievement was far greater than a simple truce. It armed evolutionary biology with a new language, transforming it from a descriptive and historical discipline into a quantitative and predictive science. The work stands as a permanent monument to the power of abstraction, a demonstration that the deepest truths of the living world can be illuminated by the spare and beautiful logic of mathematics. That this revolution was launched by a young, relatively unknown schoolteacher, working with little more than paper and intellect, makes the accomplishment all the more profound. It was the quiet thunderclap that heralded the arrival of a new, quantifiable understanding of life's magnificent diversity.